Welcome, students, to the first part of Chapter 25, which focuses on the urinary system or the renal system. So we're going to be talking a lot about the kidneys. We're going to start with kind of just the gross anatomy of the renal system, kind of what the renal system does for us. Then we'll talk about nephrons. And then in subsequent lectures, we'll talk about the actual job of the kidneys, which is to maintain the composition of our body's fluids, essentially by making urine. And then we'll also talk about how we regulate the volume and concentration of our urine, and then also how we transport, store, and of course, eliminate the urine out of the body. So what does our renal system do for us? Well, it's got several different functions. For one, it helps us regulate things like our ion concentrations. So if we're talking about like sodium and magnesium concentrations or calcium ion concentrations, the urinary system is going to help us do that. It's also going to help us regulate our water volume and thus help us regulate our blood pressure. It will also help us regulate just the amount of solutes that we have in our bodily fluids, and it will help us regulate the pH of our bodily fluids. So by excreting or kind of holding on to different acids and bases, the urinary system plays a role in keeping us at our homeostatic norms in terms of our body's pH. Since the urinary system is an excretory system, anything waste-wise that gets into the blood, the urinary system can filter out and excrete. So things like metabolic waste, like creatinine, different toxins, different drugs, right? All of that can show up in your urine. That's why urine analysis and drug tests sometimes work with the urine, because it's going to show up in the urine if it's in our body. Whilst the kidneys are not specifically part of the endocrine system, they're part of the urinary system, the kidneys do make two hormones that we've discussed before, renin, which is part of that renin-angiotensin-aldosterone mechanism that helps us regulate our blood pressure, and then erythropoietin, EPO, which is, of course, what is going to stimulate our bone marrow to make red blood cells when we have any sort of hypoxia. The kidneys can also help activate vitamin D, so plays a role in kind of calcium absorption by our digestive tract. And then, while the kidneys are not a kind of primary driver of making new sugars, if you're doing a more prolonged kind of fasting period, then the kidneys can kick in and make some new sugars for us in order to have lots of energy. So pretty much all of the work of the urinary system is done by the kidneys. See, we have two of them. Draining out of the kidneys are the two ureters. They drain into the bladder, and then the bladder is drained then by the urethra. And that's pretty much it in terms of urinary anatomy. You'll see the two renal arteries are what supply blood to the kidneys. They're quite large. And then the renal veins are what drain the blood from those kidneys. You can see the two adrenal glands sitting on top of the kidneys as well. This is the position of the kidneys. So you can see we're right around the kind of lumbar area of the vertebrae. They're sitting behind the peritoneal cavity. So this is considered a retroperitoneal position. I want to point out some of the supporting layers for the kidneys. So the kidneys themselves have a capsule around them, which helps infections that may be in the area nearby from spreading into the kidneys, because that can be really dangerous. There's also what we call the renal fascia, which is this kind of fibrous connective tissue that helps anchor the kidneys in place. And then there's this fat kind of stuffing in and around the kidneys. And one of the things that can happen sometimes with people who drastically lose a lot of weight, say through like bariatric surgery, is the fat loss occurs from that little kind of fat area around the kidneys, and the kidneys drop out of place. It's called ptosis. And when the kidneys drop out of place, it interrupts the blood flow, and then the kidneys don't work properly. So whilst bariatric surgery is sometimes quite necessary for people who have weight issues, you do have to be kind of aware of how that affects other parts of your body. This is what the kidneys look like if we were to slice one open. There are 3 main areas to the kidney. The outer region is known as the cortex. The inner region is known as the medulla. And then the very most inmost region is then the renal pelvis. You'll see that the kidneys have a little indentation. It's called the renal hilum. That hilum is where all of the blood vessels, the ureters, and the nerves will come into and exit from the kidney. The medullary layer of the kidney is broken into sections called renal pyramids. So this is the base of the renal pyramid, and at the top of the renal pyramid is something called a papilla. It's basically a little hole at the top of the renal pyramid that urine is going to kind of flow through that into the calyces. So this is a minor calyx. The minor calyces then merge into a major calyx, and then the major calyces merge into the renal pelvis. Continuous with the renal pelvis is the ureter. Again, there are 2 of them, 1 for each kidney, and the ureters are going to go to the bladder. Interrupting each of the renal pyramids are indentations of the cortical tissue called renal columns. And so you can see the cortical tissue is a different color. It's a different type of tissue. There's different things in it than what we have in the pyramids. And so they're going to look quite different. In terms of how urine flows through our kidneys, it gets made in our nephrons. I'm going to go back here for a second. All of our nephrons sit kind of up here in the cortex. So the urine is going to get made out here in the cortex and then kind of travel through these pyramids into the calyces, into the pelvis, and then into the ureter. And so the actual flow is from the nephrons into the renal pyramids, into the minor calyx. A minor calyx merges with a major calyx. The major calyces then merge with the renal pelvis. And then from the renal pelvis, the urine flows out of the kidney through the ureter, into the bladder, and then out of the body through the urethra. As I mentioned, all of the blood vessels and nerves enter and exit at that hilum, that little indentation in the kidney. The kidney is one of the few organs, like our blood vessels, that only gets innervation from the sympathetic nervous system. So there is no parasympathetic innervation. There's no parasympathetic nerves that connect to the kidneys. It's all sympathetic, branching off the renal plexus. The kidneys do have a very, very rich, strong blood supply, since their job is essentially to filter the blood. And since they're filtering the blood and adjusting the composition of the blood, they need to make sure that we're sending lots of blood there. And in fact, about every minute, a quarter of your cardiac output gets sent to your kidneys. So they are processing a lot of blood. Outside of your brain and your heart, when you are at rest, the kidneys are getting one of the largest blood flows when you're at rest than a lot of the other organs. The flow of blood into the kidney is very similar to the venous flow out of the kidney, as we've seen with many other organs as well. So if we follow that flow, we're going to, of course, be branching off of the aorta. From the aorta, we're going to go into the renal artery. The renal artery is going to branch into 2 segmental arteries. Branching off of the segmental arteries are these interlobar arteries going out through the columns, and then At the base of the pyramid, we have these arcuate arteries, and then branching out and radiating out into the cortex are the cortical radiate arteries, and you can see there's loads of them. From the cortical radiate artery, we're going to move into the afferent arteriole, which is going to bring us into the first of the capillaries of the kidney. The kidneys actually have two sets of capillaries. That first capillary is called the glomerulus. Draining from the glomerulus is an efferent arteriole. The efferent arteriole then brings the blood into the second set of capillaries that belong to the kidneys, either what we call peritubular capillaries or vasa recta capillaries, and we'll talk about kind of which ones we find where. And then we're finally kind of into the venous flow. The cortical radiate veins drain the blood out of the cortex into the arcuate veins, into the interlobar veins, and these interlobar veins drain directly into the renal vein, which will then drain into the inferior vena cava. So really the only difference between the arterial flow out and the venous flow in back is that segmental artery. There is no segmental vein here. The afferent arteriole, the glomerulus, the efferent arteriole, and then either the peritubular capillaries or the vasa recta capillaries are the blood vessels that are associated with the nephron and are part of the nephron. And we will talk about nephron anatomy separately. But like with most organs, very similar arterial and venous flow. So nephrons. Told you we'd talk about them. If the kind of workhorse of the urinary system is the kidney, well then the nephron is the workhorse of the kidney. The nephrons are what actually do all of the work of making urine and filtering the blood and changing the composition and all of those things that the kidneys do for us. It is really the nephrons that are doing it. You have 1 million of them plus per kidney, so you have lots of them. They all are out in the cortex, part of the kidney. And this is essentially what they look like if we were to look like them, kind of spread them all out. They consist of two main parts, the renal corpuscle, which is this portion here, and then the renal tubules. So here is our cortical radiate artery coming up here, right? It's radiating out to the cortex. This is these arcuate arteries. So here's that cortical radiate artery. You can see the afferent arteriole leads into the glomerular capillary, which is then surrounded by what we call the Bowman's capsule or the glomerular capsule. Draining that capillary bed is an efferent arteriole. The efferent arteriole is going to then lead into a second capillary bed. The flow of fluid through a nephron starts here in the glomerulus. That's where the filtering takes place. So then the fluid is in the capsule. Then we are in the proximal convoluted tubule. Proximal because it's close to the glomerulus, convoluted because it's all kind of complicated looking. And then we move down the descending limb of the nephron loop or the loop of Henle, up the ascending limb of the loop into the distal convoluted tubule, and then finally into a collecting duct. If we look at the kidney kind of histologically, you can see here is the glomerular capsule. So this is in the capsular space, and here is the glomerular capillaries. You can see this is proximal convoluted tubule in cross section. Here is distal convoluted tubule in cross section. You'll see the proximal convoluted tubules have lumens that are kind of fuzzier looking, and that's because they have these long little microvilli that help create more surface area. They are lacking in the distal convoluted tubules, so the distal convoluted tubules tend to have like a clearer looking lumen than the proximal tubules. So, as I mentioned, the corpuscle essentially has two parts. The capillary itself, the glomerulus, and then the capsule that surrounds it. The glomerulus is a capillary. It's a capillary bed, so it functions similarly to other capillary beds, although there are some differences. It happens to be a fenestrated capillary, so it's pretty porous. That's what is actually going to form our filtrate. It's where our filtering is going to take place at the glomerulus. And then the capsule is kind of this cup-shaped hollow structure that kind of surrounds the capillary, and essentially, whereas in most other capillaries, the capillary is just in the organ, and when the blood perfuses into the organ, it perfuses into the interstitial spaces of the organ. Here, when it perfuses out of the capillary, when that bulk flow happens and we get filtrate, the filtrate is collected by the glomerular capsule, or the Bowman's capsule. There are 2 layers to the capsule. There's a parietal layer, which is kind of the outer layer here, which is just simple squamous epithelium. And then there's an inner visceral layer that actually covers the glomerulus and has these little epithelial podocytes, foot cells essentially, that create these little filtration slits between the podocytes, essentially kind of like a sieve. And that's what allows the filtrate to kind of filter out of the blood and into the capsular space. So you can see here are the podocytes. So we're looking at the glomerular capsule. The capillaries here are kind of covered in these foot processes, and you can see these nice little filtration slits through which stuff has to kind of be able to pass in order to make it into the filtrate. Otherwise, it remains in the blood. So that's the renal corpuscle. The other portion of the nephron is the renal tubule system. Again, proximal convoluted tubule, then into the loop of Henle, or the nephron loop, and then finally the distal convoluted tubule. In the proximal convoluted tubule, there's a lot of what we call brush border, again, microvilli, little finger-like projections that help increase the surface area, because the proximal convoluted tubule does most of the reabsorption and secretion that takes place by the renal tubules. The nephron loop is mostly simple squamous epithelium, and it plays a role in allowing us to change the concentration of our urine. If you've ever noticed, when you've been dehydrated, your urine is much darker in color, whereas if you've had like a ton of water, your urine is much lighter in color. That's because your body is using the loop of Henle's to change the concentration of your urine. And then lastly, the distal convoluted tubule is more similar to the proximal convoluted tubule, but much less microvilli. So it's more of a secretory part of the tubule system than a reabsorption part of the tubule system. And then the last part of so here's up here is where we would find all of the renal corpuscles, and as well as most of the proximal and distal convoluted tubules, and then we're on into collecting ducts. So most of that renal medullary pyramids are just full of collecting ducts. And that's one of the reasons why they look so different, both histologically and kind of macro. A collecting duct gets filtrate from kind of multiple nephrons, so not just one, and then all of these collecting ducts will fuse into the minor calyces, which will then fuse into the major calyces, which will get us to the pelvis and then ultimately out of the kidney. We have 2 classes of nephrons. So I mentioned earlier we have 2 different capillary beds, right? We have 2 different types of capillaries. We have the glomerulus that all of the nephrons have, but then there were 2 other types of capillaries, peritubular or vasa recta. Whether a nephron has a peritubular capillary associated with it or a vasa recta capillary associated with it depends on which class of nephron it is. So the cortical nephrons sit really high in the cortex. Such that their loops of Henle might dip into the medulla a little bit, but not a lot. They are the ones that have the peritubular capillaries, mostly around the convoluted tubules. The juxtamedullary nephrons sit lower in the cortex. They're still sitting in the cortex, the corpuscle part of them and the tubule part of them, but they have these really long loops of Henle that dip way down into the juxtamedullary pyramids. And then you can see here that collecting duct in the pyramid as well. So juxta means next to, juxtamedullary, next to the nephron, or next to the medulla, excuse me. So 3 different types of capillaries associated with a nephron. Each nephron has 2 capillaries. They all have a glomerular capillary in that renal corpuscle that is specialized for filtration. What makes a glomerulus different than other capillary beds in the body is not only is it fed by an arteriole, that's normal, but it is also drained by an arteriole, and that is very different. So afferent arteriole leads into the glomerulus, efferent arteriole leads out of the glomerulus. Because we have 2 arterioles, not an arteriole and a venule, we have 2 high resistance vessels on both sides of the glomerulus. That makes the glomerulus a much higher pressure capillary bed than, say, your standard capillary bed in your skin or take your pick favorite organ. The other thing that contributes to this much higher glomerular pressure is that the afferent arteriole has a wider diameter than the efferent arteriole. Think like 4 lanes of traffic coming into 2 lanes of traffic. It kind of creates a back pressure. That means the glomerulus is going to be a much higher pressure system than most other capillaries in the body, which of course is going to affect our net filtration pressure. Now, the other 2 capillary types, the peritubular capillaries and the vasorectal capillaries, they are both very low pressure capillaries. They're both very porous. Both of them arise from the efferent arterioles. So they're fed by the efferent arteriole, and then they would be drained by a venule into the venous flow out of the kidney. The peritubular capillaries function primarily to reabsorb water and solutes from our urine, so make sure that we don't lose too much water and solutes. They are associated with the cortical nephrons, and then the vasorectal capillaries are found around the juxtamedullary nephrons, and they play a role in the concentration of our urine. So the peritubular capillaries tend to be, let me show you, you can see here is the glomerulus. You can see nice wide afferent arteriole and a much narrower efferent arteriole. You can see these are these peritubular capillaries. They are surrounding the tubule system of the nephron. Conversely, the vasorectal capillaries are associated with the loop part of the juxtamedullary nephrons. So you can see here is a cortical nephron, here is a juxtamedullary nephron. You can see here is our arcuate artery, then into the cortical radiate artery, afferent arteriole into the first capillary, the glomerulus, efferent arteriole coming out of the glomerulus, in this instance into a peritubular capillary, which is then will eventually drain by a venule into the cortical radiate vein, into the arcuate vein, and eventually out of the kidney. If we are a juxtamedullary nephron, again, we're going to be fed the glomerulus by an afferent arteriole. Efferent arteriole leads out, and the efferent arteriole then drains into the vasorectal capillaries, which then ultimately are drained by a venule into the cortical radiate vein, into the arcuate vein, and thus out of the kidney. You can see here is our convoluted tubules and our loops of Henle and our collecting ducts. The other bit of renal anatomy or nephron anatomy has to do with something called the JGC, or the Juxtaglomerular Complex. Again, juxta means next to, so it is this complex of structures that sits right next to the glomerulus. You can see here is your afferent arteriole leading into the glomerulus. Here's your efferent arteriole leading out. You can see a little blood in the capillary, the little filtration slits made by the podocytes. There's the proximal convoluted tubule with its microvilli. Here is our capsule. It's going to collect our filtrate. So we're looking at this part right here. We're looking at really this part right here. And so the juxtaglomerular complex consists of three different things. The macula densa, which is basically a salt sensor. It senses salt concentration in the filtrate, which is in part of the nephron loop, kind of looped up. You can see here's part of our nephron loop, kind of loops up into the near the glomerulus. We also then have these mesangial cells, which are essentially just communication cells. They've got gap junctions so that the macula densa can talk to the granular cells and vice versa. And then in the walls of the afferent arteriole are the granular cells. And the granular cells are not a chemoreceptor like the macula densa, but are essentially a mechanoreceptor. They're a blood pressure receptor. They are also the cells that produce renin, that hormone that helps us regulate our blood pressure. And we're going to talk about the role of the JGC in helping us regulate kidney filtrate formation and regulate our blood pressure through regulating blood volume. And we'll talk more about the JGC. But essentially what we're talking about is a chemoreceptor that senses salt in the macula densa and the mechanoreceptors in the granular cells that look at the blood pressure in the afferent arteriole and also secrete renin. And that is the end of urinary anatomy. So not super complicated. Now the physiology of the urinary system, a little more complicated, and we'll talk more about kind of the JGC here and the role of the glomerulus and the different parts of the tubule system in the process of making urine, because that is really the job of these nephrons, is to kind of regulate whatever solute concentration we happen to talk about, or water concentration, or blood volume, or acid-base balance. It's all happening here at the nephron.